everyone. My name is Shai Berlin. I'm from the Technion, from the Rappaport Medical School in Israel. I'd like to start by saying, first of all, this is my first time at this meeting. It's really a pleasure to see so many people that, I read their papers, I know of them, but I've never met them face to face. It's really a pleasure. I'll try to give, I actually changed my presentation last night after the roundtables because I figured, you know what, there's going to be so many people who are going to show different things and before me and give an introduction. And because this is a very, very, I would say, multidisciplinary kind of room with from parents all the way up to clinicians and so on. And so I thought I would kind of narrow it down, try to make it a little bit more focused, but also broaden and give some preliminary results that we have. And I think it would kind of be more relevant and interesting to most of you, hopefully. So a big shout out to Be Cured Foundation, who are funding part of this project. I've been working on NMDA receptors since early postdoc many years ago. And I can start by saying that we've used the GRIN 2A null mice. I have a lot of experience with them. We also see this weird, very anxious, jumping, weird behavior. And I can add a small note, when you inbreed them, they continue to have this very weird and I would say kind of a super anxious and hyperexcitable phenotype. But if you breed them out, they lose it, as one of the colleagues actually said it. And so it's a very important comment. Again, in a nutshell, NMDA receptors, beautiful receptors. They're very, very big. They span about half the size of the synaptic cleft. So, the interaction between the pre and postsynaptic neuron. Very, very big, very allosteric, very complicated. And as many people said before myself, what I would like to add and stress, because this is going to be very relevant to the next couple of slides, is the fact that NMDA receptors come in many different flavors. So we speak, we always talk, there are always 4 subunits as far as we know. There's many, many different genes that encode for these, 7 in particular. But they come in many, many different flavors in such that they can be, so always 1 is always there. The NR1, the gray, is always present in 2 copies in all receptor types throughout all development and lifetime of the individual. But the other subunit, the 2 or the 3, they kind of mix and match. I would like to bring your focus to diheteromeric and triheteromeric receptors, meaning these are receptors that are composed of 1 and 1, so 2 NR1 subunits combined with either 2A and 2B, or 2A and 2C, or 2B and 2D, and so forth. It's going to be a little bit more important once it's going to be more obvious to you why I'm stressing this out. So again, very, very challenging to actually study these, because not only do, as Andy showed before, not only do the 2A and 2B kind of switch along development, but they also switch in response to experiment and stimulus, or learning and memory formation. So again, development-wise, we have changes in in the levels of the protein levels, but we also have synaptic changes on almost a regular basis, as David Wiley described. So this is plasticity. Things change all the time. So even if we think we have a certain kind of set of receptor type and populations, they can actually change very, very drastically and very rapidly throughout as a response to some type of experience, stimulus, or just any learning and memory mechanism. Beautifully said by most of the people here working on databases. So many different variants, so many different characterization that needs to be done. I would say the first seminal paper in 2010 was kicked off this whole field, and ever since it's been really growing exponentially with the amount of different mutations. And again, taken from some of the reports of colleagues here, the 2A and the 2B, unfortunately, the most predominant receptor subtypes in the adult brain or postnatal brain, these are actually the ones that actually have the majority of the mutations or de novo mutations that we find. So it's very unfortunate because, again, these not only is 2B very important for the embryonic part, but again, 2A and 2B remain very predominant throughout lifetime. And these are actually the ones that bear most of the mutations that we find. They kind of span the entire receptor. You can see here from carboxy terminal domain all the way up to the amino terminal domain, and number-wise, in particular for the GRIN 2B. And here's just, I took from the data in the databases, and you will see that the GRIN 2B variants in particular, about 90% to this date have been completely uncharacterized. So my lab is very focused on NMDA receptors at large, but in recent years, we've really put a focus on GRIN 2B in particular, again, because of its kind of essential role in development in embryonic stages, but also because of post-embryonic and its predominance and abundance throughout lifetime. So very simple aims, very kind of reductionistic kind of concept to go towards how do we start to study these receptors. So we first of all explore how they affect receptor activity, as many of my friends here. Then we ask whether does that actually do anything to the neurons. Again, many people here describe a different phenomenon. And of course, the last question that I will hopefully present to you data about and convince you that some of the deficits can actually be rescued, as Javi just described. So I'll just give you a very brief overview of two children that we've kind of honed in from both Israel and from a Danish epilepsy center. They had a very unique non-described mutation. So it's a missense, meaning a base within the DNA was changed to another base, and instead of having, when you translate the protein, instead of having a glycine at position so and so, it doesn't really matter the number, we either get a serine or a cysteine. So for most of you, so it's not identical, meaning it's not the same, although it's the same position in the protein, it's not an identical mutation, but it's very synonymous. To the researchers in the crowd, serine and cysteine are basically the same amino acid with very small kind of, I would say, chemical changes. So, I'm sorry. So we have two patients, very young, with synonymous mutation, but they show very different clinical phenotypes. Again, not striking, because if you look at the literature, this is what happens. You can have two children with the identical mutation, but they show very different kind of developmental and intellectual disabilities and so on. And so it was curious. We need better stratification of these diseases, but this is what it is. So in a nutshell, we looked at the conservation. So we looked at this residue. This residue lies within the ligand binding domain of the receptor. So this is the part of the receptor that binds glutamate down here in orange. And it's highly conserved throughout phyla, so throughout the different organisms, all the way down to C. elegans. But trust me, we've done a bigger comparison. So very conserved within the ligand binding domain. We did some in silico modeling because we will not create, it's very difficult to create crystal structures of every single variant, but we wanted to kind of get a little bit of a kind of a gut feeling, what would this mutation do before we started screening for the actual functional effect. So in silico, they don't always work. Luckily with this mutation, it did. So what we've noticed, if we kind of zoom in to the place where glutamate binds, so the mutation is, as you can see, this is a small residue here, and it kind of coordinates the binding of glutamate. So, and if you look very, very closely, you will see that because, so again, two mutations, very, very similar, not identical, but similar. What they do is they kind of displace, hypothetically, because this is a simulation, they displace the binding of glutamate, which means, so just because I'm binding here, I'm forcing the system to bind glutamate, it does not mean that it binds, but it just shows me that there is some type of, something is going badly wrong in this very, very small and evolutionary conserved ligand binding pocket. So it almost gives you an indication that glutamate is not going to bind to that place. It has to be like the blue one, and if it's not going to happen. And indeed, if we look at, so in my lab, we have so many different preparations. We do various kinds of electrophysiology from xenopozoocytes all the way up to sheep. And this is a collection of different cell types. So what we do, we express it in different cell types. We do dose responses and so on, as most of you do as well. Again, very briefly to go through it, what we see is that it really affects the affinity or the apparent affinity of glutamate, meaning these receptors, the cyan and red here, compared to the blue, which is the wild type, they are massively shifted to the right, or in other words, they don't bind glutamate. You need 1,000 and 2,000-fold more glutamate concentrations to actually kind of nudge these receptors, meaning these receptors at the synapse, hypothetically, are completely silent. They are oblivious to neurotransmission. So the neurons can try to communicate between one another, but these variants supposedly diheteromers, so we express them as this. So 2 NR1 subunit with 2 variants, these channels, receptors are very, very severely affected, loss of function. And actually, this was the first kind of, this was the strongest severe loss of function mutation that was described for GLUN2B. The only other one that actually is as devastating is for a GLUN2A mutation, which has kind of in the same order of, if I remember correctly, it's about 4,000 to 6,000 reduction in affinity to glutamate. So very severe. Not only are they shifted to the right in terms of expression, while one subunit, again, they're very, very similar to each other, but one subunit does not affect trafficking to the membrane, and one does affect. So the G689C mutation is both devastating in terms of glutamate binding and also prevents the receptors from trafficking to the membrane. So it's a kind of a twofold reduction in the activity of the receptor. So devastating, that's all you need to remember at this point. So we said, okay, so these were heterologous expression systems. We said, what is going to happen in neurons? My lab also produces both cultured neurons. We do slices, we do in vivo. This is an example of just taking naive neurons from wild-type animals, so mice, rat and mice, the same we actually did. And what we do, we overexpress the variant. And the question is, will this variant be able to kind of mix and match within the internal machinery? Reminder, these cells already have 2 copies of the regular GRIN 2B. Again, just going very quickly across, what you can see is that AMPA receptors, so it's another kind of a sister receptor, are not very affected by this overexpression method. But if we look at the NMDA receptor responses, they disappear as expected, right? As I said, neurotransmission, if we look at the numbers for the affinity of glutamate, they should be completely oblivious. And indeed, this is what we see in the cells. So this, again, very non-physiological experiment, because we force them to express a subunit that's not supposed to be there. So it's basically to have 3 different alleles expressing GRIN 2B. But it does show that once these 2 variants express, they're very, very dominant. They predominate the system, they take over. And even if there are in the background other receptor types, maybe, we don't see them. So something very, very wrong is going on here, and it's kind of summarized here, but I won't go into the details. But this was not satisfying to us, to me, because I said, I asked myself, this is not relevant to patients. So we know that patients don't have 3 alleles of everything. And we know, and so far I've been showing you most of the data about, these diheteromers, this like very clean situation where I take the NR1 and the NR2. I put them together and I measure something. So we asked ourselves, what happens if I have a mixed diheteromer, meaning I have a regular NR2B coupled with another NR2B, but this one has a mutation? And again, what happens if I combine it with 2A? So again, mixed diheteromers versus triheteromers. We were very surprised that in the literature, there's really only a very handful of reports about this. And some of the people that have done seminal work are actually here in the crowd. So we had to start from scratch. So again, we basically have three different populations. If we look again at GRIN 2B, right, we have NR1 and 2B wild type. And we can have two variants within a receptor, and we can have a receptor that has kind of a mix. We call them pure, mixed, or wild type. Just for 2B, we used a very, very kind of a unique system developed by the Trainell's lab, Casper Hansen, back when he was a postdoc. This was a 2014 paper where they kind of do this molecular game where you can, it's kind of like Lego. You take two different parts that can only kind of assemble in a very, very particular composition, and then you can be very sure of what kind of population of receptors you have at the membrane. Should anyone want, I'll describe it later. And again, we reproduced the data that we showed before that, these mutations are devastating when they have this pure diheteromer. So it's basically the same numbers as shown before. This was just to see that, all these different molecular changes that we've done to the protein, are relevant to its function and do not affect its function. But when we put them now, when we test the different types of receptor combination, what we saw, very surprisingly, I must admit, a huge improvement in the glutamate affinity. So far I've shown you that this variant is really strongly dominant negative. It completely kills synaptic responses, but when you mix and match it with other subunits, whether it's a 2B wild type subunit or with a 2A wild type subunit, it improves fourfold the affinity. So the affinity now becomes relevant for neurotransmission. Why is this important? Why do we care about this? First of all, if it brings the concept that we can actually use drugs to modulate this receptor. If this receptor was, as shown before, completely irresponsive to neurotransmission, any drug that we would put, for example, a positive allosteric modulator would not be relevant because the channels need to open for the drug to work on. It's a modulator, it's not an opener. So before, the first experiments kind of argued against the use of positive allosteric modulators as drugs, treatments, but these new experiments actually show no, there's a huge shift, a fourfold improvement, making it relevant to be treated by different PAMs, and this is what we did. Also, for the more biophysicists in the crowd, this is very, very weird because if you think about it, so we have 4 different subunits. Each subunit has its own kind of affinity towards glutamate, and you would assume that the receptor, the complex, would be limited by the least affine subunit. So it's kind of like cars in a row. If the slowest car will kind of determine, the speed of the entire cars. And this was actually not true. So we expected the affinity to be as bad as when we have, the purely mutant receptor. And it just shows that this receptor is incredibly plastic. It's incredibly kind of modulated and allosteric. And just to be sure, one of the reviewers asked, well, is this a unique phenomenon? We collected all the different 18 papers in particular that actually described some types of di and triheteromers. It took us a while to collect it. And we actually saw that this magic number, fourfold improvement, actually holds true on average for 18 different occasions. So again, many different loss of function or gain of function mutations, when you incorporate them in these mix and match flavors, actually become relevant for treatment by different drugs. So it's good for us. Otherwise, these receptors would be completely irresponsive to many of the different drugs that we want to test on them. This was important. So brings me to the question that as triatermers are actually very abundant in the brain, if not mostly predominant in various places, such as the prefrontal cortex and hippocampus areas in the brain that are very, very important for different higher cognitive functions, now it becomes obvious and relevant to test PAMs. In a nutshell, I'll go very, very quickly. I don't know if I have enough time, but we first tested spermine. Spermine is a NR2B highly selective molecule. I know many people in the crowd here are working with different derivatives. It's a positive allosteric modulators of NR2B receptors. It has been well described. So much is known about this. I'll start by the end. Spermine completely fails to potentiate diheteromers composed of the variants, and it does not potentiate all triateromers. And this can be seen here. If you have 2B, whether it's pure or non-pure, yes, you can get potentiation, but purely diateromeric, it actually does the inverse. It inhibits. So you can imagine that if you're putting a positive allosteric modulator on top of a receptor that has loss of function, you actually worsen the phenotype by, even further inhibiting the channel. And on triheteromers, it completely fails. And again, we see very slight actually inhibition. So again, this argues against the use of spermine or other, for us, or other derivatives to treat this kind of variety of different receptor subtypes and mutations. So we stopped working on spermine, and we looked at another neurosteroid called pregnenolone sulfate. So, and in a nutshell, it's actually FDA. Well, it has been given to humans. It's fairly safe. I want to make a very strong note about safety. It does cause different side effects and symptoms, but overall, it is safe. It crosses the BBB to some extent, so it could be given orally or by other means. It's a non-specific positive allosteric modulator of 2A and 2B, so this is the best that we have. And so we said, you know what, let's see what it does. Lo and behold, it actually works well on all receptor subtypes, whether it's just the wild type or a receptor that has a good 2B and a bad 2B, two different mutations, if it's a triheteromer. And very surprisingly, if you look at kind of like a purely mutant receptor that has two variants and two NR1s, it actually works much better in terms of potentiating the current after application. Again, neurons. I don't know why you don't see the picture here, but in neurons, we said, so let's apply it onto neurons, and it does increase the overall size of the NMDA receptor current. As you can see here, in all cases, it really increases the current, meaning it works on a variety of unknown receptor subtypes. We don't know exactly on which receptor, but the overall effect is a very robust increase in the current. So that's good news. We have different drugs. We have actually in our latest paper, we describe different approaches to actually increase the amount. We can use different cholesterol derivatives and so on. And there is an FDA-approved antiviral drug that actually is now being tested because it does increase the amount of neurosteroids. So I will not get into that, but these results, I brought them here to just kind of say there is kind of hope because these positive allosteric modulators they seem to function pretty well. We just need to find the right kind of dosage and administration route. So very briefly, we said, so far, models are good, cells are good, but we need a mouse model. And unfortunately, what you can see here in this kind of summary of what we've done, so we took a mouse and we created a knock-in. So it's a heterozygote G689C knock-in mouse. You can see here by the blots. But as all my previous colleagues described, this mouse almost shows no electrophysiological, whether in culture or in slice, deficits. This was very striking, again, almost expected because of all the previous publication before ours. So neurons don't seem to mind this mutation, even though we know it's devastating in kids. We did these LTP, or long-term potentiation, as David Wiley presented before, and we looked at, at the immediate and late kind of deficits in this ability of cells to learn. So these are slices, again, so at the end, after 80, 60, 70, or 80 minutes, we see no deficits, although I can tell you that we repeatedly see some reduction in the peak response. For the electrophysiologist, we can discuss it later. I will not get into it. But again, largely, we see no major phenotype. These mice are very fertile. They reproduce pretty well. They give litters of about 8 to 10 pups at a single time. However, they're super, super anxious. We see this over and over again. The mothers are extremely anxious. They, most of the time, they actually eat their pups as well. They get this, if I'm not mistaken, David showed that the student wrote skittish or something in that. So we see that as well in these mice. It's very hard for us to kind of quantitate it because it's not very scientific to just say they're squiddish plus, plus, plus. But we see it definitely, and we're now trying to go into more anxiety tests to see whether we can see it. And again, females. In males, it's very hard to say. They're aggressive anyhow, and they're separated from the pup. So it's a little bit hard to say. And here, I'm sorry for the messy kind of slide, but the point was I just added some stuff that I asked my student to bring. Again, we do rotorod, and we do water maze, and we do open field tests. Nothing really. Maybe a little bit in the open field, maybe, at the Morris water maze, we see some deficits in their ability to learn, but nothing major, nothing very, very striking. So we're still kind of looking, and I think I got some very good kind of ideas from previous presentation, looking at some more kind of nauseous stimuli and so on that we're going to test. These mice are very, very unique in one sense because they carry the most devastating loss of function mutation described for GRIN 2B, but they don't seem to mind it that much. I'll try to conclude. So single point mutations in the ligand binding domain are devastating for receptor function. But when they're incorporated in these kind of mixed receptor populations, such as di and triheteromers, mixed and so on, they're less devastating, which is, again, good for us because, again, they start to be responsive to neurotransmission. And I forgot to say here that I'm not showing it here, but again, you can record NMDA-dependent EPSCs and AMPA EPSCs, and everything looks supposedly normal. So again, there are receptors at the synapses. They respond to neurotransmission, which is, again, whether compensation or other mechanisms are at play, we really don't know, but we've already sent out for single cell mRNA sequencing, and we're trying to do more labeling and so on. To try to understand how is this possible. And lastly, the overexpression of the variants instigates suppression of synaptic NMDA receptor activity. It really reduces the GLUNR amplitude, but it can be rescued with a neurosteroid such as pregnenolone sulfate. I'll give you kind of another kind of indication. We also see that the 2-HC cholesterol derivative also does this pretty well. And we can discuss other drugs that we've tried so far. And I'd like by thanking my lab, very, very talented lab, and of course major funding by To Be Cured. We've had some support from the European Research Council. from the Israeli Innovation Authority and the Science Foundation, from the Templeton American Foundation for very risky projects such as these kind of ASD-related diseases. From Teva Pharmaceutical, a very big pharmaceutical company that has been, that my student actually went there to try to develop a very high throughput screening, drug screening for stable cell lines expressing NMDA receptors, and the Prince Foundation for neurodegenerative diseases such as this one. And yeah, I'll end with that. Thank you.